The basic contractile unit of a muscle is the sarcomere, which extends from one Z-line to another. Two types of filaments are present: the thick, or myosin filaments, and the thin, or actin filaments. Myosin filaments are within the A band. The H zone contains only myosin filaments, whereas the I band contains only actin filaments. The actin and myosin filaments overlap within the A band on either side of the H zone. As the muscle relaxes, the actin filaments slide past the myosin filaments, decreasing the area of overlap. The sarcomere lengthens. The A band does not change. Only the H zone and I band are affected. Again, the muscle contracts, shortening the sarcomere. The actin filaments slide past the myosin filaments in the opposite direction. The area of overlap increases. Again, only the H zone and I band are affected. A closer look at the structure of myosin filaments and actin filaments will reveal how this is accomplished. The thick filaments consist of several hundred myosin molecules. Each myosin molecule consists of a tail and a head region. The tails of the myosin molecules are held together, forming a central core, while the heads are free to protrude outward. Myosin molecules are oriented with their heads toward the ends of the A bands. The thin filaments are composed primarily of actin molecules grouped together into a helix. Each actin has a myosin binding site. Tropomyosin molecules form a spiral around the actin molecules. Attached to the tropomyosin is the protein complex called troponin. Some myosin heads are attached to the actin molecules, forming cross bridges. ATP binds to the myosin head. The head changes shape and detaches from the actin. ATP is split into ADP and a phosphate. The myosin head swings up toward the actin filament. The shape of the head also changes. It again fits the myosin binding sites on the actin. Attachment to a binding site releases the ADP and the phosphate. The myosin head swivels forcefully, pulling the attached actin filament toward the center of the A band. Another ATP molecule binds to the myosin head and it detaches. This cycle is repeated many times during muscle contraction. Myosin heads attach, pull, and release. The individual myosin heads do not act in synchrony. A few are always in contact with the actin filament, preventing it from sliding back.